Here we go with the second set of video notes on our cells unit. In this one we will talk about all the different organelles you find inside animal cells. Next we'll go through some of the different organelles that you would see inside these eukaryotic cells. So first of all, the nucleus. This is probably usually considered one of the more important organelles. I know the cell membrane is pretty important too, but nucleus is usually considered the control center of the cell because it contains DNA. As you probably know, DNA is sort of like the blueprint of life. I call it life's instruction manual. It tells you all the cells what to do, basically. And the nucleus would be surrounded by another smaller membrane called the nuclear membrane, or sometimes it's also called the nuclear envelope, sort of two different terms. Some of the other features of a nucleus would be a nuclear pore. So within that nuclear membrane or nuclear envelope there's little holes that allow stuff to enter and leave. So DNA, actually not DNA, but RNA can go in and out and other things necessary to make ribosomes. And then there's also the nucleolus, which is this darker part inside the nucleus. There's a nucleolus here. Here it is in the picture. This is where ribosomes are produced. Now, it's not like the nucleolus is the nucleus of the nucleus. It's not the control center of the nucleus or anything like that. It's not where DNA is. This is just where the ribosomes are being produced. The DNA is all throughout the nucleus on the outside of that. So the DNA would be all through here. This would be the nucleolus where they're making ribosomes. So the control center of the cell is the nucleus, not the nucleolus. It's just part of the control center. The cytoplasm is obviously another big part of the cell. In a broad sense, the cytoplasm is everything between the cell membrane and the nucleus, which would include all of the organelles that we're about to talk about. So pretty much anytime you see a picture of a cell, anything between the nucleus and that cell membrane would be considered cytoplasm. The liquid part, the goo that's actually all these organelles are floating around in, that's technically called the cytosol. You will hear a lot of people when they say cytoplasm when they should be saying cytosol. It's kind of a minor detail, but it is something that we'll try to watch out for. Another super important organelle is the mitochondria. These are the powerhouse of the cell. Mitochondria being plural, mitochondrion is singular. So that's what this guy is right here. They kind of look like little jelly beans, and you see little lines in the inside of them. We'll learn a lot more about these guys later when we talk about how the cell makes energy, but these guys are the powerhouse of the cell. This is where energy is made. Ribosomes are another organelle. These are where your cells make proteins. They have a pretty important job. They are not membrane bound. Remember I said prokaryotes don't have no membrane bound organelles. These guys don't have membranes, right? And they're super small. They're really, really small. Pretty much the smallest organelle inside the cell. Another set of organelles inside the cell are types of vesicles. A vesicle is a small membrane-enclosed sac or bag that can store or transport substances. And there's a bunch of different organelles that are made out of vesicles, including the lysosomes, the ER, which stands for endoplasmic reticulum, and the Golgi, or the Golgi body, Golgi apparatus. We're going to focus on each one of those individually right now. First one being the lysosome. Right? The lysosome is a vesicle, like I just said, that contains digestive enzymes. We learned about enzymes in the last unit. They kind of break stuff down. And so this lysosome breaks down nutrients, probably first and foremost. Also would break down waste, old organelles. Sometimes an organelle just doesn't work as well as it used to anymore. And so it's got to be recycled. So that would happen in the lysosomes. Or foreign objects, if they were to, maybe if it was like a white blood cell in your body, and they would engulf a bacteria and they would need to kill it, these guys would do that. One way I kind of remember how these work is think of lysosomes are like Lysol of the cell, right? The Lysol disinfects surfaces, lysosomes disinfect your cell. They're like the garbage men of the cell. Another membrane-bound organelle is the endoplasmic reticulum, kind of a mouthful. You can say ER, that's totally fine. A lot of people just say ER and they mean endoplasmic reticulum. And the job of this organelle is to move stuff around the cell, basically. It's always located right next to the nucleus. So anytime you see a diagram, here's the nucleus, the ER is always right next to it. Here's the nucleus, here's the ER sitting right next to it. It makes vesicles, so it can make these vesicles and transport stuff throughout the cell, which is kind of like the superhighway of the cell. There's rough ER, which is covered with ribosomes. So you see over here, this guy's got lots of bumps and bumps and dots and dots. This guy has nothing. This is a rough ER. This is the smooth ER, which has no ribosomes. So the rough ER is mostly concerned with moving proteins, since that's what the ribosomes are making, and the rough ER might be moving other stuff. Another very similar looking organelle is the Golgi apparatus, sometimes called the Golgi body as well. The job of this organelle is to process and package stuff for export from the cell. So it would use vesicles to move stuff around. And so, like I said, they look very similar, so if you're ever confused by which one is which, the ER is always the one that's next to the nucleus. So this guy would move stuff to the Golgi apparatus where it gets processed. It's sort of like the post office of the cell, and then it packages up in a vesicle, and that vesicle goes along and then joins up with the cell membrane and can release the stuff. So, for example, if ribosomes on the rough ER were making a protein that the cell would need to export, they would make the protein, it would squirt it right into that ER would move it to the Golgi. As it moves through the Golgi apparatus, it might make any changes that it would need to make it more functional, and then it would create a little envelope and then move that to the outside and send it where it needs to go.